thing to note here is that there are several forms of learning and several forms of memory. They're not all single processes or phenomena. And in fact, the Spanish painter Salvador Ali painted a picture, a famous picture, which he called the persistence of memory, where he saw memory as flowing through the eons of time, not really a solid process, but a fluid process, almost like water flowing down a pipe. That's his interpretation. So, psychologists generally consider two major forms of memory. The first is called explicit memory, and that's concerned with facts and events, people, places, and things. And in theory, there's hardly any upper limit to the capacity of explicit memory. And you can't really put an upper number, ceiling on the number of explicit facts that one can learn and recall. In contrast, there's also implicit memory, such as motor learning, motor memory, which is automatic. Automatically, once it's learned and mastered, would not require a conscious effort to recall, unlike explicit memory, which does require conscious effort. And the underlying brain structures for both types of memory are shown here. With explicit memory, you've got mostly structures within the medial temporal lobes, but also within the subwhat, such as the balance involved. With implicit memory, motor learning, you've got mostly the cerebellum, a little bit of the amygdala for fear conditioning, and also the corpus striatum, which is also involved with the cerebellum in learning procedures, skills, and habits. Now, we've already touched upon memory consolidation, and consolidation is just the conversion of short-term into long-term. Whatever is important often gets mentally rehearsed over time, and the period of time that elapses during the rehearsal is needed for the conversion of short-term into long-term memory storage. And once memories are tucked away in long-term memory, most probably in areas outside of the medial temporal lobe and hippocampus, such as the neocortex, these memories can last an entire lifetime. So it's important to know that there are multiple storage sites for long-term memory in the brain, not just one storage site. And in human patients with injury to structures such as the medial temporal lobe, there are two types of memory loss that are most commonly encountered. The first type is called anterograde amnesia, whereby the ability to make new memories after the injury is completely lost. Memories obtained prior to the injury are for the most part retained. With retrograde amnesia, you get the opposite. At the time of trauma, the ability to make new memories is more or less preserved, whereas immediately before the time of injury or trauma, memories are lost. Now with retrograde amnesia, very old memories, such as childhood memories, are retained. Probably because the sites for storage are outside of the hippocampus and medial temporal lobe where injury occurs. Okay? And here's a cartoon of the human brain. That's the front, that's the back. Here's the coronal section at this level. And you can see that the hippocampi, shown here in purple, are tucked well underneath the medial temporal lobes. And are not easily accessible to experimentation, at least not to food. And one principle that we now know through anatomy, neuroanatomy, is that the hippocampus receives tons of information from multiple parts of the neocortex. frontal, parietal, temporal cortex. It does something to the information. We don't quite understand exactly what. And then it sends the processed material all the way back out to the same site of origin. So in a way, it's a loop, an information loop, from the neocortex and all the way back to the neocortex. And not surprisingly, as you'll see, the hippocampus plays a wider role than just processing spatial information because of its widespread influence on the neocortex and its collection, mass collection ability of multimodal information from many areas of the cortex. So there is a famous patient, his name was H.M. He passed away in 2009, I think, or 2008. And he's the most cited neuropsychology case in all of the neuroscience literature. He had his temporal lobes, including underlying hippocampus, surgically removed back in the 1950s because back then that was the last resort treatment for his epilepsy. He had severe epileptic seizures, which at the time could not be treated with drugs. And as a result, neurosurgeons had to remove large chunks of his medial temporal lobes on both sides of his brain. Now, upon closer examination, psychological examination, it turned out that because they took out not only medial temporal lobes, but also the underlying hippocampus on both sides, he had severe anterograde amnesia. Couldn't make the new memories for minutes, even lasting minutes after a new experience. So every time the psychologist, Brenda Milner, who's still active at McGill University, by the way, she's in there, she must be about 90, but she's still quite active. When Brenda Milner excused herself from the room and went back in a few minutes later, she always had to reintroduce herself to H.M. because he had no recollection of the earlier session just minutes prior. That's how severe his amnesia was. Everything else was okay. He had normal IQ, normal speech, sensory abilities were normal. He just could not make new long-term memories of people, places, and things. His childhood memories were intact. He could still remember the spatial layout of his childhood living room, but he could not make new long-term memories. So here's a more recent MRI taken in the 1990s of H.M.'s brain and of a normal subject's brain. You can see with H.M., these two black holes represent the missing temporal lobes, including the hippocampi. And you can see the hippocampi in the brains, in this brain of a normal human subject age max. So this was the first evidence that the hippocampus and surrounding medial temporal lobes were extremely important for making new long-term memories of people, places, and things. The case of H.M. also allowed Brenda Milner to conclude that the brain structures underlying explicit and implicit memory are not the same. Because after all, H.M. was perfectly normal in performing simple motor skills. So she could draw the important conclusion, which really led to an avalanche of subsequent research on different memory systems of the brain to make that this important conclusion. There are separate brain structures mediating these two categories of memory. Now, more recently, Eleanor McGuire and her colleagues, using functional magnetic resonance imaging of the brain, was able to show that London taxi drivers relied heavily on their right hippocampus to navigate the streets of London versus a control group of people, subjects who had no obvious spatial skills. So in most humans then, it's the right hippocampus that we use most frequently when we common with other brain functions outside of memory. There's a lateral specialization, left brain and right brain, for specific functions of the brain. This is just one example, focusing on the hippocampus. Now, there's only so much you can do in humans. You can do fMRI, but you can't really do invasive studies. So a lot of what we know about the hippocampus and its functions comes from work on rodents. And there are these classic spatial passes that we can put rodents through in order to figure out the roles of the hippocampus in spatial navigation. For instance, in some cases, the rat would be trained to remember which arms of the multiple arm maze it had visited to retrieve the food reward, so that it had to avoid visiting a previously visited arm more than once. And with practice, these rats can actually visit each arm only once and retrieve all the food rewards. So that's quite a task for a rat. So how does the hippocampus encode memory? There are experiments to show that some cells in the hippocampus are actually place cells. They fire upon the animal that is in one part of the arena, but not in another part. So in this example, they're reporting from a single hippocampal neuron in the brains of mice. And this is an aerial view of this circular enclosure. And the recording apparatus is hooked up to a computer so that the computer can plot a color code depending on how many action measures are firing as a function of space. So in this case, the animal, this particular cell fires most intensively at 2 o'clock on the animal's ear, but not as the animal is venturing into the center of the enclosure. And these are 4 examples of 4 separate place cells, each with a particular place field, a region of space in which the cell prefers to fire. So in this case, this cell's place field is concentrated here and not here. In this case, the cell's place field is right in the center of the arena. And there's no clear, obvious place field for this 4th cell. So it's tempting to hypothesize then that place cells and place fields might encode a spatial map, a brain representation of space inside our heads. And indeed, if you close your eyes, you can visualize the spatial arrangement of this room or any room that you're familiar with. That's a brain representation, a neural representation of space. And that, by the way, involves much more than just hippocampus. It also involves regions of cortex involved in spatial mapping, such as posterior parietal cortex behind the somatosensory cortex. And I'm going to try to play this. I have a video here. Okay, so that was kind of research. So here is an example of a So you guys notice that the cell fires when he's here, regardless of whether his head is pointing this way or that way. Whenever he ventures into this space, the cell fires. When he ventures outside the space, the cell is more or less silent. So that's an example of a place cell in the hippocampus. And that video was provided courtesy of Howard Eichenbaum, who works at Boston University. He's done quite a bit of work on place cells, trying to figure out what it is that they encode. Okay, so I'm back to the screen. There. So that's an example of a place cell firing. But we somehow still need to examine spatial navigation in rodents. And the most popular task was designed by Richard Morris, who's a professor of neuroscience at the University of Edinburgh in Scotland. And he designed this water maze as a specific assay for hippocampal function in rodents. And what it is about a 1 meter pool of water. The water is made milky white with milk powder so that the animal can't really see underneath. Now rodents hate water. I mean, they'd rather be on dry land. So if you put a rat around some water, it'll swim around until it finds dry land. So the whole idea is to provide a hidden escape platform, just a small rectangular platform just underneath the milky water. And position it at a fixed position, okay, somewhere in the pool. And around the pool, on the walls of the room, are placed post-visual cues, which hopefully the animal will see and use to form a spatial map so that it can find its way to the platform. So what Richard Morris has done is to show that if you lesion the cortex outside of the hippocampus, taking care to avoid sensory and motor areas, the animal still swims straight for the platform, implying normal spatial method. Control animals that have a sham lesion, again, straight for the platform. An animal with a hippocampal lesion just before training swims around more haphazardly. You see the swim pattern is a lot more random until they hit the platform. So based on this evidence and on work performed subsequently by many other groups, we now know that the hippocampus in the mammalian brain is critical for spatial learning, but also for retention of spatial learning, spatial memory in the motor space. I'm going to show you a quick video of the Morris maze for a mouse, not a rat. I like mice. They're cheap, easy to maintain, and you can mess with their genes. But here's a Morse maze. There's the hidden button. See the hidden button? These are pictures. You have Einstein and his post-relative, which not equal in intelligence. Anyway, so the experimenter will place a mouse with its head pointing this way. And what you notice is that the mouse immediately makes a 180-degree turn, apparently looks at the poster, and then it makes a straight beeline for the platform. Of course, this is a fully trained mouse. It takes about 2 weeks to train an animal to achieve this level of performance. But what's really striking is that the animal makes a 180-degree turn, faces the poster, and then it seems to make a decision and makes a straight beeline. Here it goes. There it is. Turns, looks, makes a straight beeline. That's actually quite a long distance from a mouse. There it goes. Climbs on the platform, smiles with the camera. So that's the Morris water maze, and if you were to leave the hippocampus of a mouse just before training it, it would never achieve this level of performance with the Morris test. So that's the Morris water maze. Back to the show. So there's a lot you could do with these behavioral tasks in rodents to sort of does a lot more than just encode spatial information. It's involved in a lot of non-spatial tasks, such as visual recognition. As I showed you, even verbal memorizing lists of words. And the current thinking is that when two events occur close in time together, the hippocampus is very good at making the associative links, associative memory between those two things. And with regard to spatial encoding, it's relative spatial coordinates and not absolute spatial coordinates that the hippocampus is very good at encoding. So, for example, in order for you to find your way home, you use the relative positions of key landmarks rather than memorizing their absolute street addresses. Okay? So that's what the hippocampus and areas outside the hippocampus seem to be encoding. Relative positions. For example, landmark A is to the east of B, and B is to the north of C, rather than memorizing the absolute street addresses of these landmarks, as shown here. Simply put, I don't think we have enough nerve cells and connections to save and store and remember the infinite number of absolute coordinates, street addresses. Instead, we rely mostly on relative coordinates, which makes perfect sense. So that's the essence of the water maze with the visual cues at fixed positions. When you would mess with a visual cue, change your positions around during the training, you really mess up performance. Like that. Okay, so what at the cellular level could explain memory consolidation? And I've already talked about synaptic plasticity, and nowhere in the mammalian brain is synaptic plasticity more intensively characterized than in the hippocampus. And this is because synapses in the hippocampus are very plastic. They're probably the most plastic in all of the mammalian brain. Okay. So in this simple experiment, you've got long-term depression where there's a weakening of synaptic transmission in the pathway that receives low-frequency stimulation. In the enabling pathway that receives no stimulation, you get more or less basal transmission. Okay, there's no change. Likewise, if you give much stronger stimulation, high-frequency stimulation, you get long-term potential, strengthening of transmission in the activated pathway, but not in the silent pathway, inactivated pathway. So this shows that not only are synapses plastic, but the plasticity is restricted only to the activated segment. Okay? You can't have plasticity running all over the place, even among non-specific inputs that are not activated. The key property of memory is specificity. We can distinguish one memory from another. And at the synaptic level, these forms of synaptic plasticity must or should also display specificity, pathway specificity. So these are two reasons why LTD and LTP are very good models for memory storing. They may not entirely explain memory, but they're very good models for memory storage at the synaptic level. They're very rapidly induced different memory. They last a long time after the inducing stimulus, different memory. And of course, there's pathway specific different memory types. So typically, to do LTP experiments, you can take out the hippocampus of rat or mouse. This shows the human brain, but as far as I can tell, nothing has been done. And you can cut slices, just like you would cut slices of a loaf of bread, transverse slices. Each slice is about half a millimeter in thickness. You keep the slices alive in the lab, and you can insert microelectrodes under a microscope to activate and record on specific pathways within the plants. And in my lab, we perform these recordings probably every day. In fact, my address is kind of license right now. If you want, just drop by and see the active voice experiments, something like that. And we can record for half a day, 8 hours, 9 hours, something like that, on a lot of specific inhibitors of proteins that we think may be involved in LTP. So there's a lot you can do with the in vitro preparation, and we know more now about synaptic transmission in general, thanks to work on LTP. So regardless of whether LTP or LTP, long-term depression, are ultimately proven to be the cause of memory storage, regardless of the outcome of that years from now, we still use LTP to examine the processes that enable neurons to transmit signals from one to the other. So chemical synaptic transmission as we know it today owes a lot to phenomena such as LTP and LTP. For example, some of the mechanisms for LTP expression are as follows. This is the basal condition. These are antireceptors and NMDA receptors, mostly antireceptors, which cause a certain EPSP amplitude. LTP might result from increased sensitivity because there are more antireceptors in the postsynaptic spine. So for the same amount of transmitter that's being released, one hormone, one receptor, because there are more receptors, you get greater sensitivity and a larger EPSP. You could also have a morphological change so that the neck of the spine enlarges. This reduces the resistance to electrical current flow towards the cell body for the effort that you would initiate. And that too would increase the EPSP size measured in the postsynaptic cell. You could also have an increase in transmitter release so that for the same action potential in the presynaptic cell, you now release not one, but two vesicles with the same number of antireceptors. Again, you would see a larger EPSP. So the lesson here is when we record an EPSP from the cell, we can certainly record changes in the strength of the cell, but we can't really figure out what exactly is the cause of, in this case, an increase in EPSP size. It could be one of these, and in fact, most of us now agree in the field that it's a combination of these. It's both presynaptic, increasing release, and also a postsynaptic increase in antireceptor numbers. So this is where genetics and pharmacology come into play to try to figure out which of these mechanisms apply under a certain condition. So are there any questions? All this is just a clear, right? You should get 100% when section of the midterm. Well, that's an unreasonable expectation. No, I can't speak for the other instructors. Anyhow, I think I need to reduce the volume on this. You guys can still hear me at the back there? Okay, so let's go over the first part, some of the first part, like the equilibrium potential and all that seemingly complex math. Things you should know V equals IR. More importantly, you should know the Nernst equation. can avoid using log, ln, because that's not how the exam question was set up. If you use log, sooner or later you're going to have to convert to log. So let's do some mental gymnastics. Equilibrium potential, what's the definition of equilibrium potential? It's the membrane potential Vm, at which current for an ion, that current is equal to 0. That's the equilibrium potential. Okay? Membrane potential at which the net current for a particular ion is 0. That means you can still have the ion flowing in and flowing out. It's just that there's no net current. The two currents, inward and outward, for that ion exactly cancel, or equal but opposite in magnitude. Equal in magnitude but opposite in direction. And the term reversal potential is just the equilibrium potential for the ions that cause an EPS speed snapping. That's all. So, you should know that E sodium and E potassium are at opposite ends of the scale. Highly negative for E potassium, positive for E sodium, about +60. You know, the exact values doesn't really matter. I'll tell you what the values, tell you what the values are on the exam. And ask you to choose the correct answer. There's also the equilibrium potential for chloride, which is about close to -90 millivolts. It's certainly hyperpolarized from E potassium. Okay? So you know that inward current is negative, outward current is positive. You may also wish to know that current is equal to G conductance multiplied by Vm minus En. Right? That's just Ohm's law written in the form V equals IR, I equals V over R equals equal to G times V. Okay, you don't really need to know this, but it's certainly helpful. Okay? So this term in brackets, Vm minus Ex, that's just simply called the driving force on an ion. It's basically what moves the ion across the membrane. So if this term is 0, Vm minus Ex, that turns 0, you're going to get 0 net current. I don't care what G is. G is always going to be a positive number. There's no such thing as a negative number. Okay? So this term is the driving force, and you can plug in real values to try to figure out which way the ion will flow. You can do that and plug it in, or you can simply visualize this in your mind and figure it out mentally. definition of equilibrium potential can be rephrased. It's simply the memory potential that the VM will always move towards when channels to that ion open up. So I don't care where VM is. VM could be here, could be here, could be here initially. But when channels for that ion open up, VM will always move towards the equilibrium potential for that ion, which by the way is fixed. The equilibrium potential is fixed. Okay? So if I were to tell you that VM equals minus 120 millivolts, and E chloride is equal to minus 90 millivolts. Okay? Chloride channels open up. Which way is chloride moving? Which way do you think chloride will move through the membrane? So VM is here. E chloride is up here, more depolarized, right? So when chloride channels open, VM is always going to move towards E chloride, which is more positive than VM. So to bring the memory potential more positive, which way does chloride have to go? Right?
Out.
Out. Exactly. There are infinite variations on that. You want to challenge that? Plug it in right here. Plug in VM at minus 120, minus 90, gives you minus 30, which is a negative value. Right? Minus 120, minus 90. Yeah. Gives you minus 30. So current is going to be minus. Current is going to be negative. Negative is an inward cationic current, which is an outward anionic current. So chloride goes out. So these are defined in terms of cation. Is that easy? Well, remember your nurse equation. Be able to manipulate the nurse equation. I might throw numbers at you and ask you in terms of one verb. And be sure you remember your high school rules of how to manipulate markers. It's not long. Is there anything else that you is a big puzzle question other than what's on the exam? No, it's got nothing to do with action transport. It's irrelevant. Action transport is going to be running all along. It's just as a flow of current through a particular species of channel, potassium channel, the sodium channel, voltage channel, is different. Next current. It's got nothing to do with action transport. In the living cell, action transport is always running to maintain the membrane. Strictly speaking, active transport does not occur through these types of channels. It's a good question. Is there anything else? Any questions? CNS, is there anything else you have to know? Vertical tone, sensory adaptation, yeah.
You said we didn't know the diagrams and stuff, but one thing where it's got the axon going through different parts of the nervous system, you want to see that part of the diagram?
You mean the pathways, growth or tone? Absolutely. Know those diagrams. You might be asked to reproduce them. On the short answer,
Like structure of the brain,
historically, I've never asked you to draw, I've actually never asked you to draw out the cerebral cortex or label. I consider that too anatomical. But you need to know the two pathways go for column median with the distal and spinal cord from the frontal. Where they cross. Be sure you know your separate types of sensory axons, you know, the mechanical receptors and thermal receptors. You have the A alpha, A beta, A delta, C type. Know which ones are fastest, which ones are slowest. Understand myelination, production, refractory stages. Of course, know the phases of the action potential, what causes each phase. I know the names of pharmacological blocking agents, TPS, TPA, very important. You should be able to draw out, you should be able to reproduce on paper the inward current and the outward current in the presence of TEA and TPS. So this is current, stuff like that. You should understand activation and inactivation of channels, sodium channels. You should also understand NMDA receptors. Why it's such a fascial channel. You should know LPT and long-term depression. Know stuff about visual OB column, ocular dominant column, what might explain toxicity of OB column, molecular level. You've got anti-receptors coming in and out of the membrane, etc. Let's start right now.